My name is Bailey Fadul, for those who don't know me, and I have a few announcements actually before we jump into it. But be on the lookout for an after SNR banquet drinks gathering email for grad students. So look out for that this week. And also, there was an email sent out last week of an SNR volleyball team of like grad students that we've set up too. So if you want to join that, look your email. And also, if you want to order a t-shirt, the sale ends April 11th, and that's to help with the Graduate Student Association, with us to help keep doing social events and different things that we do. So if you want to get a t-shirt, get it by Friday. Without further ado, I'm going to introduce Deepika Garugu, our speaker today, and she is a PhD student with advisor Dr. Sarah Sonstegan. And Deepika did her bachelor's from India and master's from Auburn University. Her master's program was on mollusks and arthropods, and her thesis was focused on understanding extinction risk in aquatic invertebrates using range size and genetic diversity. Now, here for her PhD, she is working on wild turkeys and in the final year of getting samples from the field. Her hobbies are hiking, bird watching, and bird photography. So she will be telling us today about the identification of landscape features that may facilitate or impede gene flow among habitat patches within wild turkeys in Nebraska and Kansas. I'll let you take it away.
Hello, everyone. Thank you, Betty, for introducing me. I'm here today to talk about one of my research objectives, which is the identification of landscape features that influence gene flow in wild turkeys across Nebraska and Kansas. So, according to a study conducted in 2019, this is the distribution of wild turkeys across United States. And in Nebraska and Kansas, we can see there is a distribution of different subspecies of wild turkeys across the states. These are 5 different subspecies of wild turkeys in United States. And in Nebraska, we can see a hybrid of eastern and Rios in mostly in the central and eastern parts of the states. And in the western part, it's mostly Merriams. And in Kansas, we have mostly Rios that are distributed across central and eastern parts of the states. And in the western part and southern parts, we have Gauld's turkeys that are mostly moved from Texas and Oklahoma as well. And coming to the habitat use of wild turkeys, they use a mixture of landscapes ranging from forests, shrublands, and grasslands, and sometimes agricultural landscapes to move across different habitat patches. And coming to the roosting sites are basically birds that use those areas to rest mostly in the night times. They use mostly hardwood trees to rest on. And coming to the nesting areas, wild turkeys are usually ground nesting birds, so they lay eggs on the ground, and they usually have like good vegetative cover that they protect themselves from predation or something like that. And wild turkeys are mostly dominantly using tall grass, mixed grass, and short grass prairie in Nebraska and Kansas. And they mostly use tree drainages to move across landscapes, that is the creeks along the tree lines, to move from one habitat patch to different habitat patches. For example, in this map, you can see this is a juvenile female bird that we caught in one of our field sites in southwest Nebraska. She moved all the way from the field site in Nebraska to Colorado, spanning over 60 miles, just to find a good nesting spot for her to lay eggs. And coming to the mating system, wild turkeys follow a polygynous mating system, which means females mate with multiple males. And they have a unique breeding system called explosive leks, where males form different groups called leks, which is usually dominated by a male, which has big beard, enormous wing spread, and they vocalize and visualize themselves through gobbling and strutting, so that they can secure like as many mates as possible. And coming to females, they usually go to these leks and try to like secure a mate so that they can have offsprings which have like all the good fitness features. And that's the reason they have like this multiple paternity in them. And here is the video of wild turkeys with the male visual displays. And that's the feature in males that is called strutting, and that is one of their things to, attract females for, secure mating opportunity. And coming to nesting and brood rearing, wild turkeys are, as said, they're ground nesting birds, and their nesting begins in the late spring and it might end in the end of July, or sometimes it can extend to mid of August as well. Once they begin nesting, they hatch between 26 to 28 days. Often, these nests are located in the ground cover, mostly in the grasslands or shrublands or forest edges. As I was saying previously, that few nests we recovered are also on the top of the rolling hills, and we saw few females choosing those kind of covers as well. After hatching, foals leave the nest within the 24 hours with the mother, and early survival of this fowls usually depends on the weather, food availability, and predation pressure. And coming to the historical distribution of wild turkeys in Nebraska, Western Nebraska usually provides a good habitat for turkeys, as they have different mixture of landscapes. Pre-1900s, wild turkeys were abundant along Niagara and Platte rivers. These are different ecoregions that are spread across Nebraska, spanning from tall grass prairie in the east with mixed grass in the center and short grass in the western part. Due to habitat loss and unregulated huntings in pre-1900s, wild turkeys were extirpated. So there were attempts made by Nebraska Game and Parks Commission in 1959, where 28 Merriams were bought from South Dakota and Wyoming to the Pine Ridge, which is the northwest region of Nebraska. And within 4 years, these numbers grew up to 3,000 individuals, and which is a good start for the reintroductions. And coming to the wild turkeys historical distribution in Kansas, Kansas is dominated with tall grass and mixed grass prairies as well, and which provided a suitable habitat for turkeys to grow. As Nebraska pre-1900s, wild turkeys were predominant across the state. And these are the ecoregions of Kansas, similar to Nebraska, spanning tall grass in the east with mixed grass in the central regions and short grass prairie in the western parts. Several rios crossed from southern border from Oklahoma and Texas and found that they made an isolated populations along the Arkansas River. The Kansas Department of Wildlife and Parks had made an initial attempts in 1959 by bringing 125 rios from Texas to southern parts of the state. This program has produced stable populations in the southern and southwest parts of the states. So using Pine Ridge as resource populations, Nebraska Game and Parks Commission has done many reintroduction programs across different parts of the states. Similarly, in Kansas as well, using the southern and southwest populations as resource populations, they have done many translocation efforts in across different parts of the state. Populations grew in diverse habitat, including the agricultural landscapes, and these wild turkeys interbred with the domestic turkeys as well. Coming to the wild turkey project, we began our project in 2020 with our first season that lasted for till mid of August. And currently we are in the third and last season of our project, and it will end in mostly July. And we have three main questions or objectives that we are working on. Firstly, the spatial ecology, it's been addressed by one of our master student who is Reese, and he's currently in the field right now. and gobbling chronology, and it is addressed by one of the other master student who is Robin, and conservation genetics, and I'm the one who is addressing questions about this. I have four main objectives. The first one is to see what factors are influencing the occurrence of extra pair paternity in wild turkeys. And the next one is to see heterozygosity fitness correlations with fitness metrics being reproductive metrics and morphological traits and disease testing as well. And the next one is to see the fine scale genetic associations during the wild turkey breeding activities. And the last one, and the one that I'm going to discuss, is identification of habitat features that are influencing gene flow across Nebraska and Kansas. So with this question, I have three main objectives to see the levels of gene flow that is happening among the sample sites, and to estimate and compare effective population sizes, and to determine which habitat features are influencing the gene flow. So coming to the capture and handling, we begin our capture in January and it lasts till mid of March sometimes. And we use walk-in and drop-off nets to capture our birds. Once captured, we go inside these traps and get hold of these birds and place them in these boxes to further process the birds. So once we take out like a bird from the box, we just take the body measurements like wing, tail, beak, fur length for mostly for the males. And each bird is equipped with the GPS transmitters and a leg band. And later we get blood drawn either from the wings or legs to have the genetic questions to be addressed. So these are the different sampling locations that we have so far till 2024. So we can see we have 5 locations in northwest Nebraska. All these are nearby to the area called Shadron. And in southwest, we have 3 sampling locations, and it's nearby to a place called McCook. Also, we've got some samples from Kansas, as Kansas is also running a wild turkey project. They have begun their project last year, and they gave us some samples to work on with, and they have different sampling sites across the state. Coming to the genetic data processing, once the blood samples are extracted from the birds, we usually store them in a freezer. After that, we follow the protocols to extract DNA and then go through the ddRAD sequencing following the protocols. And once we get the data, we have identified 5,355 loci across 338 individuals. And then to process the data to get the final genetic results, we have run through a different bioinformatic pipeline. Coming to the analysis, to identify different genetic clusters, I've used principal component analysis using AdGenet in R. So this usually helps in detecting genetic differentiations between different sampling sites, and it also helps to identify different outliers, possibly if there are any migrants or hybrids, and also to explain the patterns of gene flow or any isolation. And to identify co-ancestry, I've used fineRADstructure. It is a coalescence approach where it analyzes shared haplotypes between individuals to detect fine scale genetic relationships, and it results in identifying shared co-ancestry between the 2 individuals. So with the PCA results, we have seen different clusters of populations. With Northwest, we can see it's completely isolated when compared to other sampling sites. As it is in the northwest part of Nebraska, it was completely as isolated as expected. And southwest Nebraska is a little bit, it has a little bit connection with few sample sites, particularly with western and central Kansas sites. And coming to only Kansas sampling sites, we can see east and south have basically like some gene flow happening between them, and that's the result they have clustered into one population. And western and central have also clustered into one population. But with this, we can see there is some gene flow happening between the Kansas sites, and also some gene flow between southwest and western and central Kansas sites. To complement this, we have done fineRAD genetic structure analysis, and we have found distinct structure among different sample sites. As said, northwest stands out completely when compared to all other sampling sites, as there is little gene flow when happening when compared to other sampling sites. And with southwest, we can see some gene flow that is happening here with northwest, and some gene flow that is happening with different Kansas locations as well, suggesting there is some migration or movement that is happening between these landscapes. And coming to Kansas, we can see Kansas alone stands out again. But again, within Kansas, we can see a big block of central and west sampling sites, and east and southern, the MHKs, the southern sampling sites, suggesting that there is some gene flow with between these sampling sites, and also like between these two sampling sites with these bars of gene flow that is happening, suggesting that there is gene flow and habitat features that is promoting this gene flow between different sampling sites. And with this, we can say that wild turkeys in Kansas and Nebraska are connected through evidence of gene flow between west and central and east and south Kansas, and between south Nebraska and different sampling locations of Kansas, suggesting that there are habitat features that are promoting these turkeys to move across landscapes and promote this gene flow. To complement this, we have done landscape genomic analysis called estimated effective migration surface, which is EEMS. Both PCA and fineRADstructure analysis have revealed distinct genetic structure clusters among different populations across Nebraska and Kansas. So to see what is happening and why there is restriction between different landscapes, I have added a layer of interstate and onto the EEMS map. EEMS is usually a null model of isolation of distance. So basically it says the sampling locations or the individuals that are near together have more gene flow when compared to the individuals that are like far away from each other. So this black lines are the interstates, and with this we can say this interstate is like inhibiting the exchange of alleles between two different sampling locations, and that's the reason we can see there is limited gene flow between different Kansas sites. So coming to the connectivity conclusions from fineRAD, PCA, and EEMS, we can say there is some overlap or a gene flow that is happening between different regions. The good example is Southwest Nebraska and different Kansas sites. And they may have functional habitat corridors or patches that are promoting this gene flow and dispersal. Coming to the next steps. So with the keeping EMS as a good proof for us to move forward, I will create some resident surfaces using different environmental variables and different landscape layers. Landscape layers would be grassland, forest, elevation, and also human disturbance, including agricultural landscapes and highways. So then I will conduct a gradient forest analysis by examining how allele frequencies are changing across these environmental variables and also the landscape players that I'm going to use to enable and identify which factors are affecting this genetic variation and exchange of alleles. And yeah, thank you.
So we have plenty of time for questions. And yeah, if we have a question, can you just repeat it for people on Zoom real quick? Yeah. Yeah. What questions? Yep.
If interstates and highways are like major barriers to gene dispersal, could you potentially alleviate that by building bridges or crossing stations along these interstates? And like, in your estimate, how closely would they have to be spaced to allow gene flow between these populations?
So I mean, like, we don't know like how closely, like, for example, in northwest region of Nebraska, few of the field sites are like maybe 10 miles apart from each other, but still we see like there is mixing of these turkeys from one sampling sites to the other sampling sites. So they usually use a wide range of like habitat or landscape to move across. And I mean, like your idea of building bridges for them to move across might help to move to just find good habitat that they can, live on, survive, I guess.
Probably like get unverified and all
Yeah.
advantages.
Yeah, thank you so much for a great presentation. I have questions in terms of the research setup. How did you get your sample size and the choice for the sample size? Because on one slide you said in Nebraska you went to 5 locations and Kansas City 3 locations, and then you got so many sample size provided by the state. How was the difference in terms of sample size and what was the
So
choice?
I can speak for Nebraska because for Kansas, it's their project. And we have given them sampling tubes for them to fill in the blood. So, and sampling locations for Kansas, it's their choice. So like I'm no one to like answer them. But for Nebraska, like we chose this sampling sites mainly because Nebraska, so Northwest Nebraska has been like a good hub for wild turkeys historically, and till now there has been like the population or the numbers of wild turkeys in Northwest have been like abundant, and in Southwest there has been no studies that were done on wild turkeys ever. So to see this extreme difference in populations, we wanted to see what is affecting this turkeys to that why the population is like declining. And for each year of our project, we have to like have 60 transmitters per site, if I'm correct.
Which means 16 samples, 16
60,
That-
like we have all GPS transmitters for like 60 that
60.
No, 16, one
Yeah.
6. Okay.
60, yeah.
I just wanted to know because we have so many turkey. We're not going to all the turkey. You should have a setup or a guideline that led you to consider 16 and not 20. Or yeah, that's what I was talking about. Yeah, like the way you calculated your sample size in order to reach to 16. And then Kansas City, because I was looking at the slide, the gene, the way would you go back to the gene slide?
This one.
No, before that, yes. I can see so many samples in Kansas City compared to maybe Nebraska, there are few sample. I don't know. But looking at the slide, I can see that first of all, there is a difference in terms of sample size, but also I can clearly see that in terms of genetic makeup. Nebraska, there are two type of genes I can say in Nebraska. And in Kansas, they're all aligned, you see. Maybe the sample size might be one of the reason, but from what we see, Nebraska is totally different. And yeah, that took me to the second question, the type of genes that you were looking and what made Nebraska to be unique. And within Nebraska, we see a different setup, you know, it is a little bit isolated from the other and it's still Nebraska.
I mean, I would say that Northwest being completely isolated, first, it's far away from all other sampling locations, and also it's a different subspecies. It's Merriam's, but most of them are like maybe Rios or a hybrid of Rios and Eastern, and that could be also one of the reasons for them to be like mostly similar when compared to the Northwest ones.
Okay,
Yeah,
all right.
yeah.
Yeah, I see.
And also, with Southwest Nebraska, we had difficulty like capturing birds.
Mhm.
Mostly maybe the warm temperatures, they were not walking into the traps as we expected. And that's the reason, like maybe we have less sampling, like sample number for Southwest.
Yeah, your feature analysis will give us the answer in terms of the landscaping. Why do they prefer,
Yeah,
you know, this type of bird? Why do they prefer living here and not moving to another side? I'm glad that you put it in your future slide that you plan to analyze the landscape.
Yeah.
And we will get the answer. Yeah. That will help us to know why they prefer the grassland, what they prefer in the forest, you know, habitat type. It will help us so much. Otherwise, it's a great job, but I can see in terms of sample size, one area is many birds compared to
Yeah. I think Kansas, they got like over 300 birds last year. So yeah,
Yeah, they have a different objective
yeah, they have a different objective, yes.
in one.
I was also looking at the type of gene that you were targeting. Did you have some specific genes in the trachea that you target, or you just look at the loci?
We just look at
The loci and not specific
Maybe it's
genes.
right here. Here's a file.
I mean, I
I
didn't
think
get
he's
it.
confused. I think he's thinking a D, not a G. Not-
Oh, no, ddRAD is not like a gene, it's like a sequencing protocol that we use to, to get the sequences that we want across all the samples that we have.
So I think earlier you said they were extirpated from Nebraska and they reintroduced like 28 and that then jumped up to 3,000.
Yeah.
So do you know if like you or like previous studies have looked to see if there was like a bottleneck event and if that could have implications on like their contemporary fitness? Because I know you're kind of looking at their fitness.
In Nebraska, I did not see any studies that did what you're asking for, so yeah.
Just curious.
Yeah, even I'm interested because I don't know what's happening with these turkeys in Nebraska.
So it seems like you're kind of limited to 2 sample sites in Nebraska. Is there maybe any museum specimen or something or a reference genome that you could
Okay,
use?
so we recently got some museum samples from like, I don't know which year that it collected.
Maybe it's within the last 5, 7 years.
Okay, but yeah, we have some from Eastern, so we don't have any sampling sites in the Eastern area and few in the central part as well. So I'm excited to see how that will change this patterns.
Right?
Yeah.
Very cool.
Any other questions?
Awesome, thank you, Deepika.
Thank you so much. Thank you, everyone.
There might be some pizza left, I'm not sure, but some left way out.
Yeah, I want the
We're gonna get a rain day and then I'll stand lookout.
It's just like.
We'll have to look out for it.
I mean, brothers and sisters.